Hi, my name is Joey Castellini and I'm a researcher here at Presage Biosciences. And I'm here to share with you an example of a study that we're running here in Seattle involving local veterinary clinics and a golden named Sammy. To prepare the CIVO device, first we sterilize it and then we load it with predetermined anti-cancer agents. Each drug also is combined with a fluorescent tracking marker, which we use to both visualize the injection sites at the injection, but also the injection sites during surgery. One of the most critical steps of the injection process actually happens before the injection. We call it a pre-screen. When looking for the best injection site in a tumor, in general, what we're looking for is homogeneity within the tumor. What that really means is cellular consistency within the tumor. So we've developed our technology to be surprisingly forgiving of hitting necrotic and fluid-filled pockets, but we like to use ultrasound guidance to help us hit the best part of the tumor that we can. So once we've determined the best location of our injection on a given tumor, it's time to inject. Once the injection is done, it's all about maintaining the orientation of the injection. Now remember that we included fluorescent tracking markers in our injection contents. What's nice about the tracking markers is that it confirms that the injection was successful, and B, it confirms your orientation. And the blue light helps us determine all of these factors. Now that we've identified our injection site and our injection orientation, now we need to collect our sample. In order to do that, we pin the tumor down and we remove the unnecessary portions of the tumor. So once we've sliced the tumor, we fix it in formalin. What this means is that we bring whatever's going on inside the cells to a grinding halt, essentially freezing them in time. Later on in the lab, we can expose whatever was going on inside the cells at the time that we fixed it in formalin. Now is the time to determine what the cells are doing in response to the drugs that we injected. And the histology team does this by using a process called IHC, where they use antibodies to detect specific proteins, and then the scanner can later detect those antibodies. In order to determine how effective a drug is, we need to consider what drug we injected. If a cell responds to a particular drug in a particular way, then we get very excited.
So this image here is a cross-section of Sammy's tumor after it's been taken out off of his body. We're looking at many different things in every section and at each injection site. So first we want to know how many cells are being exposed to the drug and are responding to it. And we're looking at the number of cells that are staining positive, so in this case are staining pink. We're also looking for cells that are not dying in response to those concentrations of drugs. Those are the cells that would lead to the cancer growing back in a patient being treated with that particular drug. And one of our goals at Presage is to find combinations of drugs that, when given together, would also kill those cells. And I think it's really important for us to figure out which drugs will and which drugs will not work in a given tumor, so that we can treat the patient with the optimal drug for their tumor and avoid them all the side effects of those drugs that aren't going to do a thing in their tumor. I think that Presage has a device that will allow us to evaluate that and really solve that problem, and I'm really excited to be a part of making that happen.